Thanks for joining me. We're going to get started. That was weird. My thing took a picture. Anyways, we're going to talk about some senses today. So hang tight. All right, like I said, senses, we're going to talk about taste, smell, sight, and hearing. So we're going to start out with taste. I've kind of found that having everything drawn out helps the flow of things. So I'll do some highlighting and talk about what's going on here. Okay, so I'm going to zoom in on here. This is the tongue, right? So on the tongue, we've got our taste buds, right? And we have a whole bunch of different buds. Each taste bud has about 125 cells, okay? And these cells, they are able to regenerate about every 10 days. Okay, and so our different kinds of taste buds include salty, sour, sweet, bitter, and umami, which is like delicious or something. Salty responds to sodium, sour responds to acid, sucrose for sweet, bitter is quinones, and umami is glutamate. Okay, so we're going to talk about how the chemoreceptors work. Okay, because they're activated by chemicals. Right, these receptors are all chemical based. Okay, so for this is the salty. Whoa, that's really big. Excuse me, salty. Okay, and this is the sour. Now, these are the only two of the 5 that we have here that actually let ions into the cell. Okay, so for the salty, we have sodium that rushes into the cell. Okay, that depolarizes the cell. However, it doesn't cause an action potential inside the cell. It just causes a graded potential. Right? So then that moves down along the membrane, opens up the voltage-gated calcium channel. Calcium rushes into the cell, binds to our vesicles that we have here. Okay? And then the vesicles are going to do exocytosis. They're going to synapse with our neurons, and that's what sends the signal to our brain, and that causes an action potential in our neuron. Okay? And then that neuron takes it to the brain saying, hey, it's salty, and that's how we perceive the salty taste. Really similar thing with sour, again. We also have, sorry, I forgot to mention that we have this this channel. It's called an ENaC channel. I'm not sure what that stands for, but both of these taste buds work through this channel. Okay, so both the sodium and the hydrogen, or hydronium, whatever you want to say, ions, they both go through this ENaC channel. Okay, now for sour, the hydrogen ion comes in, kind of depolarizes the cell, but it also comes and inhibits this voltage-gated potassium channel, okay, further depolarizing the cell. But again, it only causes a graded potential, not an action potential. Okay, so then same issue, or same thing here. Opens up the voltage-gated calcium channel. Calcium rushes in, activates the snare proteins, causes exocytosis, synapses with the neuron. Neuron takes the message, the signal to the brain via action potential. Okay. Now, for your last 3, we've got the bitter, sweet, and umami. Now, these are GPCR regulated. So, G protein coupled receptor. Okay, so what we have is a ligand that comes and binds in the GPCR, right? So the sucrose, quinones, and glutamate, they're not ions, right? We see that sodium and hydrogen, those are ions. However, these 3 are molecules that they're going to come in here and bind to the GPCR. That's going to kick off the alpha subunit, and the alpha subunit is going to activate PLC. PLC stands for phospholipase C. Okay. Now, phospholipase C, once it is activated, it'll do some stuff to create IP3 or activate IP3. IP3 then comes down to the smooth ER, and then that causes a release of calcium into the cell. And then calcium does its thing, causes exocytosis of a neurotransmitter. Neurotransmitter binds onto the neuron. Neuron has an action potential and takes the signal onto the brain. Right? Nothing too crazy. So that's kind of how our taste receptors work. They're chemoreceptors, and they do this stuff. So we're going to move on to smell. Now, smell is also pretty simple, at least in the way I'm going to explain it. I'm not going to dive crazy deep into anything, okay? So I just want to point out some anatomy here. So first thing I want to point out, obviously we have our nose right here, okay? Then as you go up the nose, we have what's called this olfactory bulb, okay? And it sits just above this cribriform plate, all right? Now what's cool about this is we have little neurons that grow through this cribriform plate and go down into the olfactory epithelium, this epithelium here, okay? And then that's where, I don't know why I have olfactory neuron there, but anyways, so this is our olfactory neuron right here, growing through this cribriform plate and is open to the olfactory epithelium. What's cool is when we have a ligand, a chemical that we smell, that comes up in here, it gets dissolved in some mucus layers, and by the time it reaches here and binds to the olfactory neuron, it binds to the GPCR here, kicks off the alpha subunit. Alpha subunit activates adenylyl cyclase. Adenylyl cyclase creates CAMP or CAMP, okay? And then CAMP comes over here, and it opens up this general cation channel, okay? So we have an inrush of both sodium and calcium, okay? So that depolarizes the cell, and then also, when this happens, we open up this voltage-gated chloride channel, so then chloride rushes out of the cell, further depolarizing our neuron, sending an action potential, okay? So then the action potential goes up through the cribriform plate, and it's going to go into this glomerulus, into the mitral cell, and then it's going to head straight to the brain. It goes to two different places. We've got the temporal lobe and the frontal lobe. Now, the frontal lobe is kind of cool. That's where, when you have a certain smell that elicits an emotion or a memory, that's kind of where it is processed. So if you have good memories of walking into your home and having the smell of fresh homemade bread or cookies or some sort of a treat, or if there's a certain perfume that maybe a girlfriend, or if you're a girl, if there's a cologne that a special boyfriend or something, or husband, wife, whatever the case is, but it elicits some memories or something like that. They went to the temporal lobe, and that's where those smells are associated with memory. Kind of interesting. Okay, but what's really cool about these olfactory neurons right here is that they're the only neurons that are replaced. Okay, they get replaced every 4 to 8 weeks, so about 2 months. I just think that's really, really interesting. We are going to move on now to sight. Okay. Oh, by the way, this is another chemoreceptor, right? We have a GPCR, right? A chemical comes, binds in the ligand binding site, activating adenylyl cyclase, CAMP, all that stuff like we talked about. Okay, but again, this one is also a chemoreceptor. So taste and smell, both chemoreceptors. Now, the next thing on my list that I need to talk about, we're going to kind of skip sight here for just a second, go on to hearing. We're going to talk about how energy is carried by sound waves and how that turns into mechanical vibrations, fluid waves, chemical signals, and finally action potentials to the things we hear. Okay, so obviously we have sounds, right, that they come in. Sounds are waves, right? So we have some waves here. So waves kind of look, like this, right? So when they come down our external auditory canal, they're going to kind of cause our eardrum or our tympanic membrane to kind of bump, just every time that wave hits. Okay, and so it's going to interact with this bone here called the malleus. The malleus is going to interact with the incus. That's this bone here. And then the incus is also going to interact with the stapes. I haven't drawn it very well here, but these are they're different bones, but they're all connected. So that this stapes, as this membrane, or as the eardrum bumps, as it goes this way, the stapes hits what's called the oval window right here and causes another bump, okay? Now, the stapes, like I said, is connected to this oval window right here. Okay, oval window. And then the oval window is connected to the scala vestibuli here, okay? Now, the scala vestibuli, this is all part of the cochlea, all right? And so this cochlea is filled with fluid called perilymph right here. Okay, and both the scala vestibuli and the scala tympani, they're both filled with perilymph. Okay, now it's all actually just one big, long, continuous chamber. All right, but as soon as it reaches this point, this farthest point, it's called the helicotrema right there. Then it turns from the scala vestibuli to the scala tympani right here. Okay. Kind of confusing, but just bear with me. We're not going to get into all this stuff quite yet. I just want you to understand that as waves hit this membrane, it causes this to bump, hits this, and it causes actual waves in this fluid. Okay, and as this fluid moves through here, it is going to oscillate. There's this membrane called a basilar membrane, okay, and it's kind of a thin membrane, and it moves up and down along with the waves, okay, and as it moves this tectorial membrane, and as the tectorial membrane moves and hits on these hairs here, these hair cells, it causes a wave, and this wave moves, and it moves these hairs on what's called the inner row of hairs. These are the outer row of hairs. This is the inner row of hairs. Okay. So when this wave occurs, it bends the hairs on here. Okay. And then this inner hair cell interacts with a neuron right here. And the neuron then takes the signal to our brain saying, oh, we're hearing this frequency, right? Frequency is kind of an interesting thing. High frequency means, if we have a time period that's this long, then we have, 1, 2, 3, 4 wavelengths versus 1, 2, right? So this is a higher frequency. This is a lower frequency. Okay, and what's really cool about this cochlea is that near the front is low frequency of the cochlea, and at the very farthest point, that's I had that backwards. This is our highest frequency. This is our lowest frequency. Yeah, I think I have it right here. Here, the lowest frequency at this point. Okay, and then this is our highest frequency. So it kind of looks like this right here. If you take a look right here, as the waves come through, they vibrate here. Okay, and so if they are a high frequency wavelength like this, they're going to resonate with these higher frequency. They're basically like strings, right? But they're actually these hairs. Okay, so then it's going to register that, oh, it's a higher pitched sound Whereas once it's traveled all the way up here, it's a lower frequency and it resonates with these hairs. And it says, oh, it's a lower frequency sound. Okay, and then that all turns into action potentials, right? When it interacts with this inner hair cell. What's cool about these is they are mechanically gated. So if one of these hair cells bends this way, then it pulls on this. It's like a little chain connecting the hairs together. It pulls this chain out, opens up this gate, and then potassium rushes into the cell. As this one bends this way because this is pulled on it, then this one pulls, and again, we have another inrush of potassium, okay, causing depolarization, opens up voltage-gated channels, causes exocytosis. We have a whole bunch of neurotransmitter, goes on to our neuron, takes it to the brain. Okay, pretty cool stuff. So that's really super quick, just what's happening from sound waves to fluid waves to a chemical signal and then action potentials. So that was really quick. I'll probably come back to this and talk a little bit more about it. Now we're going to jump into sight though. Sight is really cool. It's kind of difficult to understand, but stick with me. We'll get through it. So this is super basic. There's a lot more anatomy to the eye than what I've got here. But we're just going to talk about a few simple things. Right now I want to talk about how the eye focuses light. Okay, but first it's good to understand a little bit of some anatomy. So here, this little protrusion right here, that is the part of our eye that bends light the most. Okay, it's called the cornea. All right, so we have two parts that bend light. We have this cornea, and we also have this lens. Okay, so when light is being reflected off of an object, say this is our object, it's coming, and light kind of diverges as it goes, right? So as it hits this, I should have drawn it a little different. It starts bending in, and as it hits the lens, it bends in even more. Okay, and it's doing this. Boom, boom, boom. Okay. Boom, boom, boom. All right. So that's kind of how it would look. All right. And what it's trying to do is it's focusing this reflected light through our cornea and our lens, and it's putting it on a specific spot back here on our retina. Okay. Our retina is this whole area right back here. Okay. Now, what's really cool about our lens is that it can fatten and it can lengthen, right? And it's all due to the acts of the ciliary muscle here. So we're going to come over here. I've got it drawn out a little bit. This is our ciliary muscle, this big squiggly thing, okay? In the middle is our lens. And these little squiggly guys here in the middle, those are our suspensory ligaments. Okay, they're what push and pull on the lens to cause it to fatten or lengthen. Okay, so as the ciliary muscle contracts and comes in, the suspensory ligaments loosen, right? They're not being pulled tight. So then that allows the lens shape to get kind of fat here. Boom, like this. Okay, and so that's going to happen when we're looking at something that's close to us, because we need to bend the light more than when it's far away. I don't know if you can see in my drawing here, this is a far object, this is a near object, but when this gets to this point, it has to bend it more than here. This can it doesn't change a whole ton. It's pretty straight, okay? Obviously, it's not a perfect drawing, but that is how our eyes work. The farther away something is, we don't have to bend the light, the reflected light, so much to get it to land back here on the retina. Whereas when we're looking at something close, we do have to bend it a lot more to get it in the same spot. You see, I don't know if you can tell, but it just has a bigger angle, or less of an angle, I guess. I don't know how you want to say it, but anyways, that is how our eye works, okay? So again, when the ciliary muscle contracts, kind of pushes in. Okay, these suspensory ligaments, they loosen up, and so our lens gets fat. So that when light comes in, it bends it more. Okay, now when we're looking at something far away, the ciliary muscle relaxes. Okay, and then it pulls on these suspensory ligaments. Okay, and so then it causes our lens to lengthen or to get thinner. All right, and so then light doesn't have to be reflected so much. Right, and so that's how it would look. So that's kind of how our eyes and our lens work. And what they usually do, especially if you're walking around in daylight and you're seeing colors and sharp images, whatever you're looking at directly, you're usually focusing your sight on what's called the fovea. It's a small portion, a little, like it, honestly, a pinhead-sized area on the back of your retina that's filled with cones. Now, there's two types of photoreceptors. That's what a cone is a photoreceptor. We have rods and cones. Okay, now cones are important for seeing color and detail, sharp vision. However, they're less sensitive to light. Okay, now rods, they're used to see black and white. They help us. They're the reason we have decent night vision. They're super sensitive to light, like to 1 photon of light. Rods can detect 1 photon of light. That's pretty amazing. Okay, but you don't see in great detail with your rods, and that's due to a phenomenon called convergence. So multiple rods converge on one bipolar cell, and then multiple bipolar cells converge on one ganglion. And we're going to talk about this stuff in just a second. I know it's probably not making sense right now, but it is important. So anyways, this fovea is filled only with cones, so it's made for super sharp cone images. Nothing's blocking it. Everything kind of radiates out of that spot, so it's clear, so light gets right there with no blockage, and it's what helps us see the edges and the light really well, colors and everything. And like I said, it's kind of just at this point back here on the retina that whatever you're focusing on, that's what the light is being reflected onto your fovea for the most part, so you can actually see the details. Okay, we're going to move on now to our types of photoreceptors. So I already mentioned them, right? We have the cones and the rods. There are 2 types of photoreceptors here. So I'm going to zoom in. We already talked about the cornea, ciliary muscles, lens, suspensory ligaments here, right? And all throughout our eye, we have blood vessels, right? Supplying blood to the places that we need it, right? That's always important. And on the back of our retina, we have these little dark spots that are called pigmented epithelium, and then we have our photoreceptors. So the pigmented epithelium is between all of our photoreceptors, okay, and it absorbs just errant light, okay? So we're not just being bleached out in our eyes, okay? Now, this area called the optic disc, that's where all of our nerve cells, our ganglion, exit the retina. Also, it's where all the blood flow come into the eye and leave, right? So this is also called our blind spot. The optic disc is our blind spot because there's nothing there. So this picture isn't super accurate because these are like right here. You know, they're in the way. Where actually we wouldn't have anything right here. Like I mentioned earlier, we have our photoreceptor. This could be a rod or a cone. That synapses here with what's called the bipolar cell. And then that synapses with the ganglion that takes the action potential to our brain, signaling light and giving us our sight. Okay, so we're going to move on here. Like I said, this can be a pretty difficult thing to understand, but I'm going to try to make it as simple as I possibly can. Okay, so I'm going to talk about dark first. If you're just in a dark room, what's happening on your photoreceptor? Okay, so pretty cool thing here. This is our photoreceptor. Okay, I've got it drawn out right here. In the dark, nothing is happening to our GPCR. Okay, our GPCR is blocked when it's dark. And so that means we have this channel here that's wide open. Okay, and so it's causing a depolarization of our cell, opening a voltage-gated calcium channel. Calcium's rushing in, doing its thing, causing exocytosis of the neurotransmitter called glutamate. Okay, now glutamate comes through here, and it binds to this GPCR called mGluR6. Now, when mGluR6 is activated, its alpha protein or alpha subunit pops off and deactivates what's called TRPM1. TRPM1 is a voltage-gated calcium channel. Okay, now when this is closed, there is no depolarization of the bipolar cell. So nothing happens in this cell. So then nothing happens down here. Okay, so when it's dark, this channel being opened actually shuts off everything here and here. All right, so now what happens when we introduce light to our eye? This GPCR is kind of a unique one. It doesn't have a binding site or anything like that. What happens is light comes in here, and this GPCR is called like rhodopsin or something, okay? But it's called opsin. And this opsin is sensitive to different colors, okay? Now, what's cool about this is it has this molecule called 11-cis-retinol, right? And it has this cis bond right here on the 11th carbon, okay? What happens is when light comes in, it strikes this bond, breaks it, and turns it into a trans-retinol, okay, right here. Now, when that happens, that activates our GPCR. This alpha subunit pops off, and it activates what's called PDE, or phosphodiesterase. Now, phosphodiesterase breaks up cyclic GMP. Okay, so it gets rid of cyclic GMP. Totally breaks it off. Now, cyclic GMP is what's keeping this channel open. So when cyclic GMP gets broken up, this channel is now blocked. So now we don't have an action potential, which means we don't have a voltage-gated calcium channel opening. So no calcium, which means no exocytosis, which means no glutamate in the synapse, which means no activation of mGluR6, no alpha subunit coming over here, which means TRPM1 then opens, letting calcium rush into the cell, right? Causing depolarization of the cell, which means this voltage-gated calcium channel opens up, and then that leads to exocytosis of, again, glutamate. Okay, but here on our ganglion neuron, we have a channel called AMPA, and glutamate opens AMPA. So when glutamate binds here, AMPA opens, sodium rushes into the cell, then we have, or wow, that was not good. Sodium rushes into the cell, causing an action potential, and this travels to the brain. Okay, so I know it's kind of confusing. It feels really weird, and I'm trying to explain it the best I can. So I hope that all made sense. So when light happens, it breaks the 11-cis-retinol into 11-trans-retinol, turns on PDE, which then turns off cGMP, which turns off everything else here in the photoreceptor. When the photoreceptor is basically turned off, all this stuff isn't happening, then everything else here then starts happening, because this TRPM1 opens up, we have an action potential here, and then we have an action potential here. Because this all got to be released, AMPA opened up, and we were good to go. However, when it's dark, 11-cis retinol is not changed. There is no light. Alpha subunit doesn't activate PDE. cGMP keeps this gate open. This all happens, which means this TRPM1 is inhibited, so then we have no action potentials here. I hope that made sense. But that is how we have sight, how we turn light into electrical energy. Pretty cool. We're going to talk a little bit about another phenomenon called how the center neuron or center photoreceptor responds or acts on the bipolar cell. Okay, now this is called a receptor field. Okay. So if I draw a circle here, and another circle like a target, okay, and then I draw a bunch of squares, we'll say that these are the center, and then we have a whole bunch here in the surround. Okay, I'm not going to sit here and draw tons out because I don't want to take up your time, right? But what's happening is when one of these center photoreceptors gets hit by light, if we are in an on-center off-surround then when light hits the center photoreceptor, it's going to be exactly what we saw up here. Okay? It's going to change the 11-cis retinol into 11-trans retinol. That's going to activate the alpha subunit. The alpha subunit will activate PDE. PDE will cross off cGMP. cGMP will not act, well, because it will close the sodium channel. Calcium won't be released or enter the cell. So then there's no release of glutamate. The bipolar cell will then have TRPM1 open, and that will cause a depolarization of the bipolar cell, releasing glutamate. AMPA will open, sodium will rush in, and we have our action potentials of the brain. Okay? That's just like what we talked about. Okay? Now, for the surround, however, it's the exact opposite of whatever's happening in the center. So for on center, that means if light's hitting on the center and then dark on the surround, everything again is happening the exact same, but it's just dark. So that means we have a release of glutamate. Okay? What happens here is this is called a horizontal cell. Okay? So what happens is this surround cell, because it's dark, it's releasing glutamate. And this AMPA channel on the horizontal cell is opened by glutamate. So then this cell is depolarized. It opens up this channel here that releases GABA. Now GABA is inhibitory, and it comes in through this channel here, and it inhibits glutamate release even more, right? Because we saw that when light happens on this on-center photoreceptor, we're not releasing glutamate. Now, we're still kind of releasing glutamate, right? But when we have this inhibition come through, then we release even less glutamate, which means we send an even bigger action potential. Okay, and if this site is also dark and is also sending an inhibitory response here, then we have an even bigger action potential being sent. Again, I know this is really difficult to understand. I had to work on it a lot. But just remember, these are kind of the rules, okay? Dark equals, wow, hold on. glutamate. Light equals less glutamate. Okay, this is like the main rule when you're dealing with the eyes and sight, vision. Okay, when it's dark, we have a lot of glutamate being released, right? When it's light, no glutamate. Okay, and this helps to inhibit, which means we have even more action potentials. If we have this also working in the dark, we have even bigger action potentials. Now, if this center was also in dark, then we would still have some action potentials because these are acting to inhibit, okay? But they would just have little tiny ones. So if everything here was in the dark, we would have little bits of action potentials, okay? Now, if this whole thing was in light, then we would have only action potentials coming from here, right? Because light equals less glutamate. So this would be happening, so we would have some action potentials here, but this, we wouldn't have any glutamate on either of these because they're acting basically in the opposite effect, right? So because they're not releasing glutamate, if this whole thing's in light, like I just said, then they're not helping to cause a bigger action potential. It's only coming from this guy. All right. Now there's this other thing called off-center on surround. Now it's just completely the opposite, okay? And the only difference between these two, and this is a big difference, but it's the only difference, is this channel that you have on, is this channel that you have on your bipolar cell, okay? So for on center, we have mGluR6, right? So when we have glutamate in there, mGluR6 turns off TRPM1. So then we have no action potential in the bipolar cell or the ganglia. Now, if we are talking about off center, we have AMPA, okay? And then our bipolar cell doesn't have a TRPM1. So glutamate, if you remember from above, glutamate activates AMPA, which means dark equals more glutamate. So in the dark, there's more glutamate released here. Okay, more glutamate in here in the dark. Activates AMPA, opens the channel. Sodium comes in, causes an action potential. All right. I know, it's kind of crazy. And it's difficult to understand. But that's the only difference between these two. The rules here. Light always causes less glutamate release from a photoreceptor. Receptor fields are named by what light does to the bipolar cell. So not what it does to these guys, but what it does to this guy here in the middle. So on center, light turns on the bipolar cell. Off center, darkness turns on the bipolar cell. I hope that makes sense. Like I said, this took me a lot of time, a lot of study, a lot of reading, going to tutors to figure out. But once you get it, it really makes sense. So put the time in and you'll get it. And then last little rule was surround will always be opposite of center. You'll never have on center and on surround. It'll always be on center, off surround, or off center, on surround. Okay, so those are kind of the rules here. All right, last thing I want to dive into. Thanks guys for sticking with me. We're almost done. We're going to do hearing, the mechanisms of hearing. So we've already kind of talked about it. I wanted to spend a little more time on the anatomy, right? So we have this external auditory canal. Okay, this is where the sound waves come into our ear. Okay? Again, this is our tympanic membrane, or our eardrum. As the sound waves come in, they cause this tympanic membrane to vibrate, right? At the frequency of these. They hit this, and it causes a vibration, okay? Now, this malleus, this little tiny bone, is connected to your tympanic membrane. As this vibrates, it causes movement here in the malleus. The malleus, the incus, and the stapes are all connected. Okay? So as this moves, this moves. As this moves, this moves. And then this, the stapes, is connected to the oval window. And it's kind of beating on it like a drum. So boom, boom, right? It's the same thing. Boom, boom. However, these little bones here, they serve to magnify the sound waves that are coming through, so that it has enough energy to create waves here in this perilymph. Okay, and then as we talked about a little bit earlier, these waves travel down the scala vestibuli into the scala tympani, and then they hit what's called the round window. So we have the oval window and we have the round window. Okay, so as the pressure from the stapes hits the oval window, sends the waves up, it has to have some sort of a release, because fluid can't be displaced, right? So what happens is it comes back down through here and hits the round window, and so as this comes in, this pushes out, right? And or vice versa. If this comes this way, then this means this went this way. Okay, but there's got to be a little give and take, because we can't get rid of any fluid. The fluid is always going. Okay. Now, we talked about how this is the scala vestibuli, this is the scala tympani. Now we have this middle chamber here called the cochlear duct. Okay, it's right. I've got it right here. And between the cochlear duct and between the scala tympani is the basilar membrane. All right. And then inside that, or just above the basilar membrane, is the tectorial membrane. Okay, so this is kind of a cross-sectional. I've taken a cross-section of this, opened it up. So this is the scala vestibuli. This is the scala tympani. All right, this is our tectorial membrane right here. This is our basilar membrane. And then this is the cochlear duct, this area right in here. Okay. Now what's really cool about this is the endolymph, what's inside the cochlear duct, is high in potassium. So then as the waves come through this scala vestibuli and causes some vibration through here, we get some movement of these hair cells, right? And like I talked about earlier, as one of these hair cells gets bent, it pulls on this chain, opens up the gate, and then potassium rushes into the cell, depolarizing the cell, right? And then as it does that, what's cool about these outer layer hairs is that they have this protein on the inside called prestin. And what prestin does is when this potassium comes in, it activates this prestin, and prestin starts to compress and like push and pull. Okay, so it goes up and down. It goes boo, 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 boo. Right? Causes even more vibration, so it amplifies the waves that are already coming through here even more. Okay, and so as this all starts to go up and down like this, it creates these waves, and it comes and it pushes on these hairs here on the inner hair cell. And then, like we talked about earlier, this inner hair cell is what talks to the brain. It synapses with the neuron, sending what we hear to the brain. Okay. I don't really want to get too much into why it's so high in potassium, but it is important to remember that your endolymph is high in potassium, your perilymph is high in sodium. Okay? And yeah, I think that's all we're going to talk about, because otherwise it could get really complex. Our hearing is quite the subject. So to just cover it again really quickly, and try to make it as simple as possible to understand, we have these waves, auditory vibrations, that come into our auditory canal, vibrate the tympanic membrane, which cause movement in the malleus, incus, and stapes, which then hit on the over window, causing actual waves to occur in our perilymph. These waves transfer through everything here, causing movement of the tectorial membrane, the basilar membrane, and these hairs. As these hairs get pushed and pulled, potassium comes in, causes Preston to help with the movement, even more amplifying the signal. As this all gets moving, causes waves to come here and activate our inner hair cell, which, when potassium rushes into this cell and brings calcium in, then we activate an action potential that leads to the brain, and thus we hear. So that's all we're going to cover. That is everything for our senses. That was a lot of information. Thanks for sticking with me, guys.